microorganisms living with us, and because most of them are not disease-causing agents. On the other hand, there's a few that are. So what's immunity? We're going to be talking about both innate immunity and adaptive or acquired immunity tonight, if we get that far. Immunity can be defined as resistance to or ability to resist disease, infection, and disease. So there's a difference between infection and disease. Here we see some people maybe afflicted. That little guy in the middle definitely afflicted with something. I think that's me. Anyway, so what is infection? Infection is when you have bacteria colonizing some part of the body where they don't belong. So we have lots of microorganisms that live inside our alimentary canal. They're in the gut. Those organisms belong there. That's their normal space to be. But if those organisms get into your bloodstream, that's not normal. So they're colonizing, or they might be colonizing, some place where they don't belong. Then you have an infection. Sometimes they can get into the bloodstream for a very short period of time. For example, if you have your teeth cleaned, oftentimes there will be just a little bit of damage to the gums and some black bacteria get into the bloodstream. They may not actually colonize. They're going to be taken out by your immune system fairly quickly. Disease exists when there's physiological and anatomical changes that result from microbes present, and those changes cause impaired function or dysfunction such that the overall health of the individual is negatively impacted. So damage is being done. We're going to get to eventually mechanisms of pathogenicity, and we're going to find out some of the things that microbes can do to cause damage. But you can't necessarily say these things are going to happen automatically. You get exposed to the microbes, and then there's a contest between them and your defense mechanisms and your normal flora and so forth. For this class, we're interested in infectious diseases. So infectious diseases are those that involve microorganisms. We're not going to be looking at diseases resulting from, say, vitamin deficiency or intoxication because you've been drinking lead-tainted water or you've been taking mercury compounds. Now, our defense against infection and disease is maintained in part by our immune system, but we're going to find out immunity is dependent upon a variety of things, and exactly what the immune system is varies depending on whose book you're looking at. Lots of different cells and tissues are involved, and for a long time, immunology was not included in physiology textbooks because there wasn't a specific organ that could be identified as being the immune system organ. And now we know there's a lot that the microbes that live in the gut play an important role in immunology in helping us defend ourselves. So it's fairly complex, this immune system. The normal microbiota, I'm going to talk specifically about what those guys do. It's hard to decide where to put them because technically they're not really part of either our innate immune system or our adaptive immune system. They're organisms that colonize us very early in life, and yet they do play a tremendous role in immunity. As I said, some physiology textbooks are now considering the gut microbiome to be an organ. We just don't know what they're doing. So they're working on that. The other thing about immunity is that humans vary considerably in terms of what kind of an immune system they have. So for this class, we're going to assume that we're talking about a normal, fully functional individual. Some people have compromised immune systems for various reasons, sometimes genetically. They just don't have the immune system they should. We're going to assume that we're talking about a normal, healthy, functional individual and present all aspects of immunity. This is kind of like when I introduced eukaryotic cells, I said we're going to talk about all eukaryotic cell structures, even though a lot of organisms don't have chloroplasts. Our cells included, we don't have chloroplasts. Well, so there's some normal, healthy, functioning individuals. A lot of different lifestyles. People can interact with various aspects of the environment and protect themselves pretty well. Anyway, so again, for convenience, we're going to divide immune mechanisms into two categories, innate and what is called adaptive or acquired. For most of the time, I'm going to refer to the adaptive or acquired immune system as adaptive. It is acquired in the sense that it requires that you encounter some type of foreign agent. If you didn't, somebody, maybe your mother did at some point. Anyway, the other difference is that innate immunity is non-specific, whereas adaptive or acquired immunity is very specific, and we'll see why. So we're going to talk about innate immunity first. Innate immunity is that which is built in. So you have immunity at the time of birth. That means that the cells involved in this are, for the most part, already present. Again, there's some exceptions to this, because if we want to include the normal microbiota as part of your innate immune system, technically you pick them up during the birth process. You're passing through the birth canal. So yes, you pick them up before you actually come out into the external environment. But anyway, they get on you. So innate immunity, again, provides non-specific defense. What that means is that you have a wide range of potential pathogens that is going to defend you against. And so it's not really specific, it's not against a particular type. So the cells, the tissues, the immune substances associated here are quite varied. And again, for a long time, not included in physiology textbooks because they weren't seen as a system per se. They still may not be. A lot of times, if you read about the immune system proper, you're going to be reading about adaptive immunity and not innate. But we need to talk about innate immunity anyway. So the other thing is sometimes you'll see our defense against foreign agents described as first line of defense, second line of defense, third line of defense, etc. And it gets kind of shaky because not all references are the same, although pretty much we can say that our first line of defense is skin and mucous membranes. So we're talking about innate immunity. The first barrier that's going to prevent foreign agents to gaining access to our cells, our tissues, is our skin. Skin and mucous membranes. This is going to provide a barrier between us and the external environment. So our first line of defense against potential pathogens. This is my very simplified view of a human body. You look at that and go, wait a minute, that kind of looks like a pink hamburger with some funny looking shapes on it. But the dry skin covers all of our external surfaces. So the outside portions of the curve, that would be where we have dry skin. And then the internal structures that you see are respiratory system, gastrointestinal system, urogenital system, and those all have mucous membranes. So when we say dry skin and mucous membranes, it's dry skin on the outside and it's mucous membranes on the insides of certain systems. So skin and mucous membranes, we can say that these provide two types of barrier. One is physical and one is chemical. Physical barriers are also called mechanical barriers. And it's important to recognize the difference between physical and chemical. Again, on, was it the midterm or this last quiz? Some people had some trouble with that. Now, it was on the midterm because I was asking about a chemical mutagen, and a bunch of people told me ultraviolet light. It's like, ultraviolet light is not chemical. That's a physical mutagen. Anyway, so know the difference. So mechanical aspects, these are physical aspects of skin that make it an effective barrier against potential pathogens. Number one, the skin is multi-layered. So if you take an anatomy class, you're going to end up learning about the layers. There's an epidermis on the surface, and under that there's a dermis. But then when you look at that more closely, you find out even those layers are more complex. The epidermis, our epidermis is made up of stratified squamous epithelium. Stratified means multiple layers. They're kind of flat on the surface, so these cells are being produced down in here. The epithelial cells are produced here, they move up toward the surface, and as they move toward the surface, they change in shape, and they also change in their composition. So they're highly keratinized. Keratin is a protein associated with these surface cells, and it makes them fairly tough. Keratin is also insoluble. Watch for spelling here. In fact, this whole lecture tonight, I'm going to be giving you guys information about terms that look like other terms that you have to be careful about. So we use the term keratin or carotenoid. We talked about carotenoids. Those were the pigments, and it was spelled C-A-R-O-T. The carotenoid pigments were like carrots, orange or yellow. This is completely different. This is keratin spelled with a K, and it's K-E-R. Anyway, keratin, this is the protein that makes the skin surface tough. You have much more keratin in certain regions, like on the palms of the hands or the soles of the feet. So it's that tough layer, and in the picture in red, you see those fibers. The other thing about the surface cells is that they're dead. They're dead, and consequently, as the new cells come up underneath them, those top cells come off. They're constantly being shed. We got to see this in the laboratory because you took a toothpick and just gently scraped the inside of your cheek. That would have been a mucous membrane, and what you scraped off were epithelial cells. Those would have been epithelial cells stained with methylene blue, and we see the bacteria on the surface. Well, so the cells are coming off, and they're like rafts with a bunch of bacteria sitting on them. They come off, and so as they're shed, bacteria are shed along with them. The other thing about those surface cells, again, they're dead, they're constantly being shed, being replaced by cells that are coming up underneath. They form kind of like a multi-layered residence hall for bacteria. The bacteria on the surface, but they're also underneath those layers where you can see the separation between the cells. Now, underneath the epidermis is the dermis, and the dermis is composed of dense connective tissue. So oftentimes you'll read about the dermis as being a leather-like barrier, but what's leather? Skin. So yeah, very leather-like, because leather is the dermis. Most of those fibers are collagen. Collagen proteins have a lot of strength, and so this is going to make that dermis a very tough layer. This is showing some samples of leather. You can see the collagen fibers on the surface of that one, but this is going to form a very tough layer that is going to resist breaking and tearing, and it makes it hard for microbes to get in. There are also chemical aspects of dry skin. So the chemical aspects, one, the surface of the skin is salty. This is because we perspire. Perspiration contains salt and water. As the water evaporates, the salt is left over. So here you can see some images. You can see perspiration on the surface of the skin. Its function is cooling. As it evaporates, that cools the skin surface, but then more is going to be produced, and it leaves behind a layer of salt. So on this t-shirt, you can see salt. This person was sweating, and what's left when the moisture dries is a layer of salt. Consequently, the skin surface is quite salty. If you're working outside in the summertime, stop, check out the surface of your skin, you'll find out, yep, definitely salty. The skin surface is also acidic. pH is about 5.5. Neutral pH is around 7. So 5.5 is pretty acidic. When we grow bacteria in here, most of our media are between, the pH is between 6.8 and 7.2. So they really want to be at a neutral pH. 5.5 is definitely acidic for them. They're not going to like that. So the salt and the acid together creates an environment that's pretty hostile for most microbes. They're not going to grow readily. The other thing that happens is on the surface of the skin, there's oils and waxes. These are produced primarily to maintain the skin itself, that is to keep the skin healthy, but it also provides waterproofing, and it can inhibit microbial growth. On the other hand, some types of microbes can get down into some of those glands and cause problems, so they get down into hair follicles. Now, we've looked at skin, both mechanical aspects and chemical aspects. Now we look at mucous membranes. Mucous membranes also have mechanical aspects, that is, they're multi-layered, but they typically don't have as many layers as the dry surfaces. And the layers are different. So the mucous membranes always have an epithelial layer. You take anatomy, you find out that not all the epithelium is the same. So sometimes it's stratified squamous epithelium, sometimes it's pseudostratified columnar epithelium, sometimes it's transitional epithelium. There's a lot of different types. And underneath that, there's a connective tissue layer, but it's nowhere near as thick as the dermis. Mucous membranes are often covered with mucus. That's a lovely, lovely photograph. I don't know if it's like, who takes pictures of their kids that look like that, but anyway. Well, so mucus has a number of functions. It's sticky, so it tends to trap particles that might otherwise just pass by. If it's in the nasal passages and so forth, you breathe in a lot of microbes in the air. We saw that on our air plates. Those typically don't get into the lungs because they're stuck in the nasal passages. They get stuck in the mucus. So they never get down much below that. In certain parts of the body, mucus also provides lubrication. So in the mouth, you mix it with the food that comes in so you can swallow readily. And then it gets into the stomach. Food and some microbes get into the stomach. The mucus there is protecting the stomach from the acids that are secreted. Although there are some bacteria that can live in the stomach, most of them can't. Stomach has an exceedingly low pH, sometimes 1 or 2. but that's not a mechanical aspect. We'll come back to that. Some mucous membranes are equipped with cilia, and the respiratory system is a good example because it has cilia, and the cilia are mostly moving. They're going to be able to move pathogens up and out of the airways. So they're creating currents that move things. There used to be a lab in physiology where you would open up a frog, and you could put a little tiny piece of, we call it pith. It was a material that comes out of a plant stem. You put it right on the surface and actually watch the cilia move it. And it'll move along very rapidly. So the cilia create currents that move things up and out of the airways. Here you can see some images of cilia. And then typically what they do is move particles into the pharynx. The pharynx is an intersection between the respiratory system and the gastrointestinal system. Things get in there, and then you swallow them, and they end up in the stomach, which is, again, extremely acidic pH, and there most of them die. This is a little short movie showing mucociliary clearance of particulate material. So it's going to talk about what cilia do, how they work.
Every day we breathe in more than 15,000 liters of air, enough to fill around 1,600 balloons. Even in the most pristine environments, that air is not clean and pure. We inhale thousands of dust, smoke, and virus particles, about 100 bacteria alone every minute. More than 150,000 contaminants each day that, if given free rein, can infect and clog the respiratory system. These foreign particles, viruses and bacteria, are trapped in an extremely sticky mucus layer lining the airways. Mucus carries the trapped material into the larynx in a process known as mucociliary clearance. The physiology of this clearance process isn't completely understood yet. Research continues, but let's take a close look at what we do know. We know that mucus transport to clear foreign particles is driven by motile cilia. Cilia are small tentacle-like structures with a diameter about 1,000 times smaller than human hair, which beat in an asymmetric rhythm. Cilia protrude from most of the epithelial cells lining the airways, densely carpeting the respiratory tract. This was revealed by scanned electron microscope images.
Now these cilia would be moving particles to the right.
Size is indicated by this scale. The cilia are bathed in a watery fluid. The mucus layer, which traps debris, floats on top of this periciliary fluid, and as the cilia beat synchronously, the mucus layer moves, carrying along trapped particles. Now, the exact mechanism by which the cilia cause the mucus to move remains unclear, and it's the subject of intense research. This particular animation is based on the work of Gaber and Pratt.
And those cilia are moving in slow motion, because they go a lot faster than that.
The cilia are shown here in slow motion. In a healthy person, they beat much faster, typically around 15 cycles every second.
There we go. Really moving.
Propelling the mucus about 10 millimeters every minute. Zooming out to a video microscopy recording of live tracheal tissue, you can see the tiny beating cilia as flicker throughout the image, and debris floating on the mucus layer as dark spots moving slowly across the field.
But see which direction they're going? Now they're going to the left. It's like first they show you a film where everything's going to the right, now they're showing you a film and everything's going to the left. So they just reversed it. But they do move particles fairly quickly. The other thing that's kind of interesting is that smokers damage their cilia significantly. If you need a reason to not smoke, that's a good one. Because you don't want to get rid of the cilia that are moving microbes up and out of your airways. And apparently cigarette smoke kills those guys. Or at least decreases their efficiency significantly. Okay, so those were mechanical aspects of mucous membranes. Now we'll look at some chemical aspects. And mucus itself plays a dual role because it is part of the mechanical aspect, catching things and allowing them to be moved, but it's also a chemical. Now mucus and other secretions such as tears and saliva contain enzymes called lysozyme enzymes, and lysozyme will hydrolyze peptidoglycan. So this is an enzyme that is specifically going to cause damage to bacteria, particularly gram-positive bacteria that don't have a membrane outside their peptidoglycan layer. So lysozyme will break down peptidoglycan, basically killing bacteria. It breaks the bonds between the N-acetylmuramic acid and N-acetylglucosamine. It's like, oh, you have to go back and think about peptidoglycan structure. The other thing about lysosine is it acts as an opsonin. Opsonization is a feature that I will talk about when we get to adaptive or acquired immunity. Basically what it means is it makes particles attracted to phagocytes. So if lysozyme binds to bacteria, not only does the lysozyme damage the bacterial walls, but it can cause phagocytes to come after the bacteria and catch them and consume them. The other thing about mucous membranes is oftentimes mucous membranes have antibodies associated with them.
I'll never clear my throat again. I know it could be something. I'll never say food. I'm sorry. Following it. At least
That may not bother you guys, but it drives me nuts. Okay, so mucous membranes have antibodies associated with them. Now, antibodies are definitely part of the adaptive or acquired immune system. We're going to see overlap between the two systems, and this is one of them, where antibodies are associated with mucous membranes. They're actually attached to mucous membranes. These are antibodies in the isotype IgA. Come back to isotypes later. So some of these are attached to the membranes. Some of them are actually released into various secretions. Again, not just the mucus in your mouth, the saliva, but also tears and also breast milk. Mucous membranes sometimes have a very low pH. 5.5 on the skin, the external surface of the skin, is considerably more acidic than considerably more acidic than neutral, the 7.0. But the stomach can be as low as 1 or 2. So this is an extremely acidic pH, and within the vagina, the pH is typically 3.8 to 4.5. Again, quite acidic. This is going to restrict what types of organisms can live there. Stomach acid kills most types of bacteria. We just had an outbreak type though on the quiz this week, Helicobacter pylori. Those guys are adapted to living in an extremely acidic environment, and they do live in the stomach. Then there are phagocytic white blood cells. Now, this is typically considered our second line of defense. Phagocytic white blood cells are going to be encountered once a pathogen has gotten past dry skin or gotten past mucous membranes and has gotten into the bloodstream. Phagocytic white blood cells. White blood cells are called leukocytes. Leuko means white, cyte means cell. So be careful about this because leukocyte is spelled somewhat similarly to lymphocyte. A lymphocyte is a specific type of leukocyte. Not all leukocytes are lymphocytes. Anyway, so this is our second line of defense. We know that lots of types of eukaryotic cells can take in materials such as viruses through endocytosis or pinocytosis, if you will. But there are certain types of white blood cells that can actually take in other cells. These are referred to as phagocytes. So if they're phagocytic white blood cells, they're capable of taking in particles much bigger than just viruses. They can take in bacteria, and they tend to consume large numbers of bacteria. These phagocytic white blood cells are divided into two categories when they're in the bloodstream. They're either called monocytes or they're called neutrophils. Unfortunately, those names have lots of variations, and so a monocyte, depending on where it is, can be called something else, and a neutrophil has more than one name. Well, okay, phagocytosis, remember it means cell eating. That means the particles that are being taken in are cellular in size. This cell that you see here is a monocyte. This is a very large phagocytic white blood cell. After they take materials in, those materials are enclosed in a vesicle or vacuole, and that's what you can see in the cytoplasm. These cells typically have a blue-colored cytoplasm. These white spaces are the phagocytic vesicles or food vacuoles. This is the nucleus. The nucleus is supposed to be horseshoe-shaped or kidney-shaped. It's quite variable. The shape of these cells overall is quite variable because they often will make pseudopodia, big long extensions that make them anything but spherical. So eventually they're going to digest these materials. They're enclosed in food vacuoles, which are then brought into contact with lysosomes. and the food would be broken down by lysozyme enzymes, or sometimes they might be in lysosome enzymes, which are hydrolases, or they might be broken down within peroxisomes. All of that's kind of review stuff. So monocytes, that's what that picture was. The monocytes are large. They're called agranular leukocytes or agranulocytes because they don't have granules in their cytoplasm. Their cytoplasm is just blue. They form somewhere between 3 and 8% of the white blood cells in the circulation. We're going to be looking at blood smears. You'll have an opportunity to see the phagocytic white blood cells. Monocytes are fairly easy to find. So again, they're big, blue-colored cytoplasm with these white-colored food vacuoles or food vesicles present. When they leave the bloodstream, which they do, these things undergo a process called diapedesis. They go through between the cells of the blood vessel wall and they get into the tissue spaces. Recall the film that I showed you guys about a eukaryotic cell where we took, it started out showing you the inside of a blood vessel, and then it took you inside the eukaryotic cell and you got to see a lot of things happening. And at the end of the film, what you saw was another view of the white blood cell coming along, attaching to the inside of the blood vessel wall, changing shape and scooting in between this vessel, the cells that make up the vessel wall. Anyway, that process is called diapedesis, and most of the white blood cells that are produced don't stay in the bloodstream. Our bodies actually produce more white blood cells than red, and yet our blood is red because we have a lot more red blood cells in the circulation, and it's because the white ones don't stay there. They go out through the tissue, or they go out into the tissue, and then they change their name. The term histiocyte, histio just means tissue. So a white blood cell that becomes a histiocyte was a monocyte, now it's a histiocyte. That's just a generic name. It means, okay, it's in the tissue. Well, depending on where it goes, it can have a different name. Another name for monocyte is macrophage. Macro meaning big. These are the largest of the phagocytic white blood cells. But they go to some location and then they can have a different name. So if you were studying hematology, you want to know where are you finding them, and therefore what would you call them? They go into the liver, we call them Kupffer cells. If they go into the lymphatic tissue, we call them dendritic macrophages. And you don't have to know that stuff. You just have to know monocytes that also are sometimes called macrophages. They're these very large phagocytic white blood cells. But look at the pseudopodia on this thing. So they form these big, long extensions of the cytoplasm, and this one's grabbing bacteria, these are grabbing bacteria. They grab those and then bring them up and take them in through phagocytosis. Again, phagocytosis is cell eating. They're taking in big particles. So these things will typically ingest cells that are coated with opsonizing proteins. The opsonizing proteins, one of them is associated with the innate immune system, substances that we're going to see in just a minute that are called complement. And the other opsonizing proteins are antibodies. Well, antibodies are associated with adaptive or acquired immune system. So you get to hear about both, maybe tonight, maybe not. Anyway, they can also recognize certain types of pathogens by means of these pattern recognition receptors that are called toll-like receptors. I'm not going to ask you about toll-like receptors either. You can get into the specifics of innate immunity way beyond what I'm presenting here, and it's very complex. Anyway, they have to be able to recognize that which they should eat versus that which they should not. So if you have very large numbers of monocytes in the bloodstream, this is a condition called monocytosis. Too many monocytes, and it is indicative of a disease state, usually indicating that there's some kind of chronic infection. Bacteria or viruses have been there for a while. So if you wonder why do you have a blood test, what are they looking for? They might be looking at the number of a certain cell type, and if they're looking at monocytes, monocytosis means there's too many of those guys in there. So here we see this condition. You should not have this many monocytes visible in the same field of view. That's way too many, so that's monocytosis. Again, indicating usually a chronic disease state. The second type of white blood cell is called a neutrophil, but these things have nuclei that vary. They can have 3, 4, 5, 6 lobes. Sometimes the nucleus looks like a worm. Consequently, their nucleus is polymorphic, and so they are called polymorphonuclear leukocytes, which is abbreviated PMN. Sometimes people call them PMN, sometimes they call them polys, short for polymorphonuclear leukocyte. These are granular leukocytes or granulocytes, which means they have granules in their cytoplasm, but their granules don't show very much because they don't take up the stain. When you stain blood cells, there's a stain called the Wright stain that is used, and neutrophils don't take up the stain, so you don't see those granules. Anyway, these guys are very abundant. They make up about 70% of the circulating white blood cells. When we look at blood smears, it's really easy to find neutrophils. They're all over the place. The background on these guys, the ones on the left-hand side are darker than they usually would be. Usually they're just kind of a pale lavender, and you can see their nucleus as a dark staining body that has a lot of different shapes. So these are smaller, significantly smaller than monocytes, but they can still engage in phagocytosis. They also engage in diapedesis, so they leave the bloodstream and go out into the tissue spaces. And here we see neutrophils increasing. So there's a bunch of these guys. They've moved into a space where bacteria have gotten into the tissue, which is something that they'll tend to do. And they display positive chemotaxis. Positive chemotaxis, that should come across as something you know, right? Positive means they're moving towards something. Chemotaxis means they're being stimulated by chemicals. So they will tend to be attracted by chemicals such as interleukins and interferons, gamma interferon, and complement factors. So we had this little film we watched once before.
Neutrophils are white blood cells that hunt and kill bacteria. In this spread, a neutrophil is seen in the midst of red blood cells. Staphylococcus aureus bacteria have been added. The small clump of bacteria release a chemoattractant that is sensed by the neutrophil. The neutrophil becomes polarized and starts chasing the bacteria. The bacteria, bounced around by thermal energy, move in a random path, seeming to avoid their predator. Eventually, the neutrophil catches up with the bacteria and engulfs them by phagocytosis.
I love the sound effects. Slurp those guys in. Anyway, you saw that film once before. It's like, seriously? These guys can also increase in number when there's something going on. They typically suggest that there's some type of acute bacterial infection. So if you have too many neutrophils, it's neutrophilia. Something's happening, bacteria have gotten in. Acute means something that's short term. So these guys increase in number very quickly. And here we see some examples of tissues that have neutrophilia going on, and this might be something that would stimulate that kind of a response. So there's an acute situation happening there, inflammation. In that case, it looks like a carbuncle probably. Might be staph infection. Okay, so we had our first line of defense, we had our second line of defense. Now we can talk about some other aspects of innate immunity that I don't want to put numbers on. We call that first, second, third. It's like, no, we're not going there. So, also associated with the innate immune system are tissues called reticuloendothelial tissues. Reticulo means net-like. So if you look at these tissues, there's a certain type of fibrous material that can be stained, and it looks like a bunch of fibers extending across the tissue. In association with these fibers are endothelial cells. Endothelial cells are the same kind of cells that are found on the inside of the heart and on the inside of blood vessels. They look like epithelial cells, but they're inside, so they're flat. Typically, they arrange themselves in a single layer, but in the endothelial tissue, they're kind of wrapped around these fibers. So you have fibers with endothelial cells on the surface, and between those are spaces. These tissues then are somewhat sponge-like, and they have very specific functions within the body. Certain types of tissues that are reticuloendothelial tissues are the liver, the spleen, lymph nodes, the thymus gland. There's a variety of little patches of reticuloendothelial tissue. There's some associated with the GI tract called Peyer's patches. There's some in other places. So one of the features about them, these reticuloendothelial tissues, is they typically have lymphocytes associated with them. Lymphocytes are the primary players in adaptive or acquired immunity, and this is significant because these reticuloendothelial tissues will filter blood or lymph. As these body fluids go through, then whatever kind of pathogens might be filtered out can be taken in by phagocytic white blood cells that also live there. So here we see some white blood cells in lymphatic tissue. Actually, that's liver tissue because those are Kupffer cells. Kupffer cells arise from monocytes that have left the bloodstream and got into the tissue. If we look at the two major functions of reticuloendothelial tissues, one is filtering. As I said, fluids pass through these, either blood or lymph, and so they act like a filter. Materials are going to be stuck on one side, and then the phagocytic white blood cells that are present can readily consume those phagocytes. Well, our phagocytic white blood cells are what we call antigen-presenting cells. So what they'll do is take in a foreign agent, they digest it, and then they present on their cell surfaces some of the components of those particles that they've consumed, and those components then are recognized by T cells, and they also can stimulate the production of more lymphocytes. So these reticuloendothelial tissues allow for very rapid communication between the phagocytes that are consuming the pathogens and these other types of white blood cells, lymphocytes, that are part of the adaptive or acquired immune system. We'll talk specifically about what are the lymphocytes, the T lymphocytes and the B lymphocytes, and the roles that they play. So communication between innate immune system and adaptive or acquired immune system is going to happen largely in these reticuloendothelial tissues. This is why they're so important. For a long time, physicians would remove tonsils when they became swollen, and after a while somebody said, you know, those tissues are probably swollen for a reason. They're actually doing something, some kind of physiological process is taking place there. Maybe it wasn't such a good idea to just rip those out. And now they don't. No, you should not just rip your tonsils out. That's not okay. Another type of innate immune response is inflammation, or an inflammatory response. This is very complex. So again, we're looking at just kind of the surface of inflammation. Inflammation is characterized by an increase in redness, swelling, and temperature within an area of traumatized tissue. Redness and swelling and temperature all have Latin alternate words. So you might read about inflammation as being an increase in rubor, which is redness, tumor, which is swelling, or calor, which is an increase in temperature. They're all going to happen. The area of traumatized tissue, what that means is some kind of damage has occurred and probably some type of pathogen has been introduced. Please don't talk during presentations. It's very disruptive. And it also suggests you're not really paying attention, which you need to do. So this increase in redness, swelling, and temperature is also usually accompanied by pain. The swelling puts pressure on nerve endings, so that's going to hurt. So an inflammatory response involves a lot of things. Kind of a simplistic view. Again, we take it step by step and say, okay, this has to happen and then this happens. These traumatized tissues, the cells that are involved are called mast cells, at least some of the cells. Mast cells contain an inflammatory substance called histamine. And when damage happens, the mast cells degranulate, which means they release these granules and histamine goes into the tissue. They also contain prostaglandins, but much of the inflammatory response has to do with histamine by itself. So it turns out these cells release these inflammatory substances into the surrounding fluid. Histamine is what we call a powerful vasodilatory substance. It's going to cause vasodilation. Specifically, what it dilates is arterioles. Arterioles are the blood vessels in front of capillary beds, so they're going to increase the blood flow into capillary beds. The human body has, I want to say, 70,000 miles of capillaries. I wonder who measured that. Like, come on. But anyway, an average human, 70,000 miles of capillaries. Most of them are closed most of the time. And during an inflammatory response, they fill up with blood. So blood goes into them. This is going to account for the increase in redness and to some extent the increase in swelling. Along with this dilation that allows more blood flow into the capillaries, you also get an increase in permeability of the capillary wall. If you increase permeability, that makes it easier for things to go through, and some proteins that would normally be inside the blood vessels will go into the tissue spaces. When the proteins go, liquid follows. Typically, the water goes into the tissue space also, and you get swelling. Now, to add more to that, the prostaglandins, which are also released by those mast cells, prostaglandins cause vasoconstriction. But they cause vasoconstriction, so you have arteriole and then capillary bed and then venule. If you constrict the venule and you've opened the arteriole, you've got more blood in the capillary, you've closed off the exit, and so the pressure is going to cause even more fluid to go out of the capillary bed into the tissue, and again, you're going to get swelling. The prostaglandins also increase capillary permeability, so it all kind of works together. So we have damage, trauma, swelling. That specific region, you can see quite a bit of swelling there. And as I said, this is often warm to the touch. If you've had a child running around barefoot in the summertime, they run around on a, say, a redwood deck, they get a sliver. Maybe you don't see this response right away, but if you don't know that sliver's there, 2 or 3 days later you find it and it's like, oh, the bottom of the foot is swollen, it's warm to the touch, it's red. That's an inflammatory response. And all these things are going on. So this increase in capillary permeability, coupled with the increase in hydrostatic pressure. Again, hydrostatic pressure is fluid pressure because you're constricting the venule, you've opened the arteriole, you're getting more blood into the capillary beds. All of this is going to cause fluid to go into the tissue space, and so we get this swelling. And again, proteins and other kinds of, well, materials that are in the bloodstream are going to go into the tissue space. Some of those proteins are antibodies. They're part of our adaptive immune system. Some of those proteins are complement factors. Some of the proteins are fibrin. Fibrin has to do, it's going to be involved in clotting responses, and complement is an opsonate, among other things. Anyway, so we get a bunch of proteins going into the tissue space, and then water follows, and all of this is going to cause the swelling. Now, there are some other substances called leukotrienes that are released when the white blood cells. One thing that happens if you increase capillary permeability, it makes it easier for the white blood cells to go through and get into the tissue space. They are attracted by chemicals that are released by those traumatized tissues, so they will go in there. It's really easy. The leukotrienes attract them. It's easier for them to go through the vessel wall, so they get into that area. And then the white blood cells themselves begin to release things. So they get called into the area, and then they eat some bacteria. They consume those guys and get rid of them, but they also produce lysozyme. As I said, lysozyme kills them. So they start killing the bacteria once they didn't eat. And then there are antibodies that come into the area. Those are some of the proteins that are in the bloodstream, primarily IgG and IgM, and they're going to have a whole bunch of different effects that we'll learn about when we talk about adaptive immunity. The white blood cells also produce complement. So complement is another protein that's produced and goes into the tissue space. This is by itself part of the innate immune response. So complement is part of the inflammatory response, but complement kind of gets recognized as being an innate immune response by itself. You have these proteins, the complement proteins, they're typically numbered 1 through 9, and that's how they're identified. So you have C1, 2, 3, 4. C1 through 9, they cause opsonization, which means they can stick to the surface of bacteria or other types of material and cause phagocytic white blood cells to want to consume those particles. You can think of opsonization as making things more tasty, more attractive to phagocytes. The other thing that complement factors do is they make holes in cell membranes. So they work in a series. You'll sometimes hear about complement fixing, and what that means is one type of complement binds, and then it splits in half, and then that triggers a series of responses where other complement proteins interact, and they make holes in cell membranes. We're still talking about some of these substances that are released in association with inflammation, so we'll come back to complement in a minute. Another substance that's released is called pyrogen. P-Y-R-O, pyro, means temperature elevating. So things that are going to generate an increase in temperature or fever. And there are exogenous pyrogens. So these are substances typically released by the pathogens, the organisms that got in association with the damage. These are oftentimes lipopolysaccharides associated with gram-negative cell walls. They're in that outer membrane. Or they can be endogenous pyrogen. Endogenous pyrogen is a substance called interleukin, which is produced by monocytes and macrophages, and it causes fever, but it's kind of fever overall. So the exogenous pyrogens tend to cause an increase in temperature in an immediate area, whereas the endogenous pyrogens tend to go they act on your thermoregulatory center in the medulla and cause an increase in temperature overall. So you get fever. Oh, hypothalamus, sorry. Back the truck up. Not the medulla. The hypothalamus has your thermoregulatory center. We go back and study the brain. Okay. So a major feature of inflammation, trauma occurs, you're going to increase blood flow to the area, but again, why do we want to increase blood flow to the area? Well, one major reason is to get the phagocytic white blood cells in there, because they will consume bacteria and they can also kill them with lysozyme. So here we see it, that looks like a pin stabbing into that tissue, but it might have been a splinter. Some bacteria get in, and even after the damaging splinter or pin has been removed, there's still bacteria left behind, and then you get this response of phagocytes coming in. Phagocytes eat bacteria, they also kill them with lysozyme, and they release complement factors, which can make holes in them, and they release endogenous pyrogen. Or they'll cause the release of endogenous pyrogen. So here we see complement. These are the complement factors here. This combination, C6-7, actually it's 5-6-7, they form what's called the membrane attack complex. And then these C9s literally make a hole. Make holes in the cell membrane, water can go through, and the cell will lyse. So antibodies are also going to come into the area. We get phagocytes in, but we get antibodies in. They can cause opsonization. They can do a lot of other things, which we'll learn more about. The other thing that happens is fibrin. The fibrin comes in, and fibrin is part of our mechanism to build clots. There's a series of events that happen to get fibrin into the area, and then it kind of walls off the area and prevents the pathogens from spreading into the bloodstream. So they kind of have to stay put. And there we see kind of a localized effect. Some kind of damage occurred. We have a lot of phagocytes come into the area. They're consuming bacteria. They're killing bacteria. You get this accumulation of dead white blood cells and dead bacteria, and this is pus. The term for pus generating is pyogenic, P-Y-O-genic. So be careful about pyrogenic versus pyogenic. Words that look very similar, but rather different. Pyogenic is pus forming. And our body elevating temperature, that's an attempt to kill pathogens. So our body temperature will go up in response to pathogens getting in as a way to kill them off. Now, back to our innate immune proteins. So we're still talking about the innate immune system. There are several different proteins that are part of innate immunity. Some I've mentioned already, but I want to talk about them in a little greater detail. So there are interferons. The term interferon comes from the fact that these proteins were found to interfere with the life cycles of cytolytic viruses. So there were certain cells within the human body that would release interferon, and certain interferons would then go to neighboring cells, and viruses couldn't get into those cells. So they couldn't get in, they couldn't complete their life cycle. It was thought for a long time that the interferons were going to be a very effective antiviral type chemical. Unfortunately, what was determined after a period of time is there isn't just one type of interferon. There's a whole bunch of different interferons, and they have different effects, and even the same one can have different effects on different people. So this is showing type 1 interferons that are interferon alpha and interferon beta. They have antiviral action. They can also act against tumor cells. So they do have some specific effects, and sometimes these are used clinically to treat certain situations. Type 2 interferon, what's called interferon gamma or gamma interferon, this is something that attracts phagocytes to an area and also increases their activity. So if gamma interferon is being released, then more phagocytes come into the area. They'll start eating bacteria and digesting them. The idea, initially when interferons were discovered and people recognized they had some antiviral activity, they thought, well, there's genes that code for the proteins, the interferon proteins. How about if we put those genes into E. coli and get E. coli to produce interferon? So they did, and E. coli do make interferons. As I said, they have different effects on different people. So sometimes they work pretty well, sometimes not so well. A lot of it depends on individual variation. Another type of innate protein are called interleukins. These are referred to as cytokines. Cytokine just refers to a substance that's produced by a eukaryotic cell and typically has a high molecular weight. So these are released by lymphocytes, they're released by macrophages, they're also released by a variety of other cells. For a while they were called lymphokines because people thought they were only released by lymphocytes, but they're not. They're released by a variety of cells. So the interleukins, one of these is endogenous pyrogen, so it acts on the hypothalamus to cause changes in your temperature regulation so that you will elevate your temperature overall. They're going to cause fever. There's other interleukins, 2, 4, 5. We're going to talk about a type of interleukin associated with adaptive immunity that stimulates cell proliferation. So these interleukins have a lot of different functions. And again, this is kind of beyond the scope of this class. A group of innate proteins I really want you to know about, again, are the complement factors. So these complement factors, they're serum proteins, but they're produced by macrophages. And they get into the circulation. When they activate, they become proteases. So they act on one another. The first one will act, and then that will cause the next one to act, and so forth. So they act in a sequence. They can, there's certain ones that will create what's called the complement cascade. This is showing you a representation of a complement cascade. Typically this cascade starts with C3 and then goes in different directions. So you have all these different complement factors that get involved. Obviously you're not going to know that thing, but the classic pathway, C3 is cut in half, you get C3A, C3B. C3A causes opsonization, so it's going to stick to things like bacterial cells and make them attractive to phagocytes. Meanwhile, the C3b goes off and causes this membrane attack complex formation. So we get other complement factors. We get the membrane attack complex, which is C7, 8, 9. The 9s actually go into the membrane and make the hole. So this is showing C5, C6, and then 7 and 8 is this little guy down here. C9 is the one that really makes this hole in there. So basically complement factors, they can cause opsonization, and they make holes in cell membranes. Unfortunately, they're not entirely specific about what kind of cells they make holes in. The good news is our bodies, our cells, have factors on their surfaces to prevent complement from being activated. So most of the time, the complement cascade doesn't occur, but there are situations, we're going to talk about hypersensitivity, allergic reactions, where complement can be activated on human cell surfaces and make holes in them, and then they die, which is not okay. It's supposed to be a defensive type substance, complement. This is showing the type of cells they destroy, which are red blood cells. If you get complement activation on the surface of a red blood cell, it'll blow it up. Another factor that can be included in innate immunity is a type of cell called a natural killer cell. I'm never quite sure where to put these guys, and I think I mentioned them here and also in association with adaptive immunity. But they are innate in the sense that they're not specific. So innate immunity is non-specific. These guys kill other cells, and they're not terribly specific or selective about which ones they kill. So they aren't lymphocytes. They're not considered part of the adaptive or acquired immune system because they're not specific. They have substances inside them called perforin and granzyme. The granzymes are proteases, and both of these can kill other cells. Again, they're not terribly specific about what cells they kill. So the perforin makes holes in cell membranes. The granzymes destroy cells from the inside. If this was a cell that was infected by virus, it would not only kill the cell, but it would destroy the virus particles that were being made in there. So it's a very effective defense mechanism. So the granzymes degrade proteins inside the targeted cells. They kill the cells by a process called apoptosis, where they are degraded and they blow up. Like I said, if that was a cell infected by virus, it'll not only kill the cell, it'll destroy the viruses too. Well, and then we have our normal microbiota. They're kind of on the borderline. They're on the edge of our adaptive or acquired immune system, and they're on the edge of the innate immune system. We acquire them during the birth process. We've talked about these quite a bit this semester. So we have mostly bacteria and fungi. But we also have some protozoa that live inside our bodies. We also have some multicellular type organisms that live in association with our bodies, and a lot of times we don't like to think about it. We have these funny little mites that live in the follicles associated with our eyelashes. Apparently they're always there. You'd hardly notice. Anyway, so our normal microbiota become associated with us fairly early in life, and although they change a little bit, they tend to stay remarkably similar over time. Now there's some suggestion you can actually identify people that were involved in a specific incident by looking at kind of fingerprints left behind by traces of their normal microbiota, which is really weird, but anyway. So what do? Well, they provide protection by competing against potential pathogens for available nutrients. So we've seen situations where you're growing bacteria on the surface of an agar plate. You know that when colonies are very close together, they don't get very big. This is because bacteria will obtain nutrients even outside the border of their colony. If you have a lot of colonies, they can't get big because there isn't enough nutrients. They run out of food. So if you have a bunch of normally present organisms that are taking up most of the nutrient, this is going to tend to inhibit pathogens from growing. They can't get the food they need. The other thing they do is they compete against pathogens for binding sites on cell surfaces. This isn't something we've talked too much about relative to bacteria, but you know that viruses have to bind to their host cell surface in order to infect. Well, bacteria do also. They have to bind, and that binding requires a specific receptor site. So we have bacteria that tend to infect certain regions of the body. That's where the receptors are. If those receptors are covered up by normal organisms, the ones that are normally present, your normal microbiota, then the pathogens can't even get a foothold. The other thing that these guys do is they produce various types of chemical substances that kill other cells. One group of substances are called bacteriocins. The bacteriocins tend to kill only closely related cells. So your E. coli will produce substances called colicins that kill other E. coli. But you have Staphylococcus on your surface, on the skin surface, Staphylococcus lugdunensis. They produce a substance called lugdunate that kills Staphylococcus aureus. So they can kill sometimes organisms in the same species, but sometimes organisms in a different species. Now, new studies suggest that disease processes involve creating imbalances within these microbial communities. You're supposed to have a certain number of organisms and a certain variety of organisms, and if you don't, you develop what's called dysbiosis. Dysbiosis is imbalance, where things are out of whack within your microbial communities, and this is going to allow pathogens to get a foothold, to get established, and begin to cause disease. So we know that our normal microbiome is responsible for a variety of metabolic processes. As I said, they're in your gut. They're not just sitting there. We know they produce vitamins that we use. They also help us digest our food, but new evidence shows that's not all they do. They're associated with immune development. You don't have proper gut microbiota, you don't develop an immune system properly. And they even influence brain activity, so they can influence the behavior of an organism and may be associated with certain types of disease states. So be nice to your normal microbiota. You can't get along without those guys. Okay, so that's innate immunity in a nutshell. And now we'll see how far we get into adaptive or acquired immunity. So we're still talking about host defense mechanisms, but we're talking about the immune system proper, adaptive or acquired immunity. So again, this is defense against infection and disease, but it's specific defense. It's unlike innate. Innate, like your skin surface, is protecting you from everything that's out there. Your adaptive or acquired immune system is protecting you against very specific things. We say that this is acquired because it involves some type of interaction with a pathogen previous to the response being initiated. It doesn't necessarily have to happen in the individual, however, because it could be there was a response that happened in the mother, and then that immunity can be passed to the offspring. So we say that specific immunity is adaptive, and I'm actually going to call this adaptive immunity. It is acquired by some previous exposure, but it's definitely adaptive because what happens is you have cells within your body that get genetically modified. So we typically think of all of our cells having the same genetic complement. We know that we have so many chromosomes and that the composition of those chromosomes should be set. All those cells come from one. But when you start looking at the adaptive or acquired immune system, it's adaptive because it's actually modified. Cells that are going to give rise to antibodies are genetically modified. Certain portions of genes are removed. So it's kind of like post-transcriptional modification, but post-transcriptional modification was modifying RNA, and this is modifying DNA. So what happens is you get exposed to a pathogen sometime previously, and then if that same pathogen gets into the body a second or subsequent time, then your adaptive immune system is going to respond to that particular pathogen. We'll see what kind of responses. The other thing that could be said about this system, although we say it's acquired immunity, the cells that are involved are definitely built in. So the cells are present at birth, but those cells change. The cells go through a series of processing steps, and they become specialized so that they can respond to a particular pathogen. So their response is acquired. We're going to have interaction between human cells and specific pathogens. Again, it doesn't necessarily occur in a single person because it can occur in a mother and then be passed to an infant, either prior to birth or passed to an infant through breast milk. Well, so this is acquired immunity. I want to talk about how is it acquired. So we can think about this immunity as being active. It's active if the body is stimulated to produce its own immune cells and substances. The cells involved are lymphocytes. We're going to talk about B lymphocytes and T lymphocytes. But if it's active immunity, we're actually going to stimulate the production of those cells and some substances released by those cells. If it's passive immunity, then the body is not going to be induced to produce its own immune substances. You're going to get immune substances from somewhere else. Notice the differences in color: active immunity in yellow, passive immunity in blue. The other way that we can categorize how you acquire this immunity is whether that acquisition is natural or artificial. It's natural if it involves some type of mechanism available to living organisms in general, and it doesn't make any difference whether you live in a technologically advanced civilization or not. If it's natural immunity, anybody could be exposed to it. If it's artificial immunity, then it's dependent upon how much technology is available. Do you live in a society that can produce vaccines, for example, or can they provide you with immune serum? Notice again color difference. Natural is green, artificial is pink. Completely arbitrary, but anyway. So adaptive or specific immunity can be acquired in 4 ways. This is kind of like our nutritional categories where you had energy categories and you had carbon categories, and when we put them together, this is like that. We've got natural and passive, we've got active and passive, natural and artificial. So if a person encounters a virulent pathogen, they get sick by being exposed to that pathogen, and if they're lucky enough to respond, they can become immune. So we're showing this in the images. Somebody's sneezing, those viruses get passed to somebody else, that person gets sick, and then they're going to develop some kind of response, and then they're good. If they're lucky, they have immunity. Well, so this is natural because they're exposed to the pathogen under natural circumstances. Anybody could have been exposed, and they are induced to produce their own immune cells or substances, so it is active. We've got green and yellow. Another way that you can acquire immunity is to receive immune substances. You can be given antibodies or cytokines that are coming to you from your mother. If they're coming to you from your mother through the placenta, everybody has a mother and a placenta at some point in time, even if you have a developing embryo that's in a container, at some point it's going to get put into a uterus, it's going to have a placenta, and so it can get antibodies that way. And then after birth, if an infant is breastfed, they can get antibodies from breast milk. So here we see placenta, and we see some little infant having some breast milk. So this immunity is natural. The individual is acquiring natural immunity. from the mother, either through the placenta or through breast milk. But the individual is getting pre-made antibody, pre-made immune substances, not being induced to produce its own, so it's passive. It's natural acquisition, but passive. Third way is an individual can receive a vaccine. Vaccination is the process. Vaccine is the substance that is administered. If you're given a vaccine, then you will be induced to produce your own immune cells and substances, and then you can become immune to a pathogen. So this is artificially induced, and the body is being induced to produce its own immune cells and substances, so it's artificial and active. And then the fourth way, you can receive pre-made immune substances, pre-made antibody from some other source. Sometimes this is produced in another type of animal. Sometimes antibodies are grown in tissue culture. They have what's called monoclonal antibody that's produced by cells that are basically cancer cells. They are immortal, so they just keep growing, and they can be induced to produce a particular type of antibody over a long period of time. So this is acquisition that is dependent upon technology that makes it artificial, and because you're not asking the body to do any work by itself, it's passive. Okay. Now, for simplicity, from here on, I'm not going to talk about the adaptive acquired immune system as adaptive and acquired. I'm just going to call it adaptive, or I'll try to. Anyway, so adaptive immunity from this point on, unless I forget. The primary cells involved are lymphocytes. These are agranular leukocytes, white blood cells. They were called lymphocytes because they were associated with lymph. They also increase in number within lymphatic tissues. So lymph-type cells, lymphocytes. There we see some examples of lymphocytes. These are smaller than monocytes, but agranular, again, so their cytoplasm is blue. And they come from the same type of cells that produce monocytes, the agranular leukocytes. These cells all come from what's called hematopoietic stem cells. And that word hematopoietic, if it's spelled with an A, that's a European spelling. If it's at the beginning, hemo or hematic. If it's not, then it's American. Anyway, these are produced by cells that are in the red bone marrow. Turns out all lymphocytes come from the same types of cells, and then they differentiate, and you get different types of them. There are multiple different categories of lymphocytes. I already mentioned the NK cells, the natural killer cells. That's one category of lymphocytes. But there's two that are associated with adaptive immunity. Those are T cells and B cells. So B lymphocytes are B cells, T lymphocytes are T cells. The NK cells, the natural killer cells, used to be references said they were neither T nor B. Other references I've seen say they are some kind of modified T cell. They are most similar to cytotoxic T cells, but not as specific. They used to be called null cells. Anyway, as I mentioned, they have perforin and granzyme. We already talked about them, so never mind. Now we'll talk about B cells. Where did the name come from? Why are they called B cells? Well, it turns out that back in the 1960s, in birds, there was an organ, a specific organ that was called the bursa of Fabricius. And cells that were going to become B cells had to go into that tissue, and they got differentiated. And when they came out of that tissue, they were B cells. So then everybody thought, well, humans must have a bursa equivalent. And they started looking for the bursa equivalent tissue. As far as I know, they never found it. Consequently, now if you look up what the B stands for, it stands for bone marrow. Well, that's handy, except all blood cells come from bone marrow, so it's not very specific. But they do come from the red bone marrow. You can see images there. Red bone marrow exists only in certain regions within the skeleton, inside flat bones, irregular bones, and the proximal ends of long bones. That's where you find the red marrow, and that's where these hematopoietic stem cells come from. The B cells have to go through multiple stages of development. Exactly where they do this, I don't think is understood, and maybe it's multiple different tissues. Again, their genetic content is actually modified. So certain genes get sections taken out, and you end up putting together slightly different variations, and as a consequence, you can get lots of subpopulations of B cells that are a little bit different from one another. Here we see some stages of B cell development. You can find lots of images online, and it gets rather complex, again, beyond the scope of this book. this presentation. So, cells that in the lecture notes I refer to as immunocompetent B cells, apparently there's been some name changes. Immunocompetent B cells are now referred to as naive B cells. If they're naive, that means they haven't yet come in contact with a specific antigen that they need to respond to in the future. So you have all these subpopulations of naive B cells, and at some point they have to encounter an antigen, and then they're going to be programmed to respond to a particular antigen. They will do this under the influence of helper T cells, and we'll talk about helper T cells. So that's a different category of lymphocyte, the helper T cell. They're going to release substances that are going to cause certain populations of B cells to reproduce in large number. Some of those are going to become B memory cells, and others are going to become plasma cells. So basically what you see going on here is you've got the T cell, this is probably a phagocyte actually grabbing something. Phagocytes will present antigenic determinant groups on their cell surface. The helper T cell recognizes those antigenic determinants, releases substance that tells the B cell to proliferate, make a bunch more. Some of those B cells become plasma cells and they produce antibody and release it. And some of them become B memory cells that can stay in the body for an indefinite period of time. And we'll talk about why that's significant also in just a bit. Or maybe not today, but anyway. So you have all these different subpopulations of B cells. Why does it matter? Because you're exposed to different antigens at different times. And what the T cells do is cause certain subpopulations of B cells to reproduce in very large numbers. You have to have a lot of B cells reproducing to make enough antibody to do any good. So here we see this accumulation of naive B cells. Each one has on its surface receptors. They actually have antibodies on their surface that can bind with specific antigens. So this antigen gets in, it can interact with the antibodies on the surface of this particular cell. That particular cell is then going to be stimulated to reproduce. You get a whole bunch of cells that can produce antibody against that specific antigen. This is what we mean by adaptive immunity. So you have the subpopulations of B cells, but only certain ones of those subpopulations are going to be stimulated to reproduce in large quantity and produce enough antibody to defend you. Now, so that's B cells. Come from the bone marrow, but as I said, all white blood cells do. The T cells, the T stands for thymus derived. So T cells also come from the red bone marrow initially, that is the T cell precursors, and then they go into the thymus gland where they get processed into specific types of T cells. So the thymus gland, be careful of this one. This section has a whole bunch of terms that sound kind of like something you might know. Thymus, not thyroid. In the throat region, you have a thyroid gland. Below that, kind of in the upper chest, much lower in the throat, is a thymus gland. Different kind of spelling, different kind of gland. The thymus gland is something that's fairly large in young individuals, and then it decreases in size as you get older to the point where it's almost not there at all. And the thymus gland is where these T cells get processed. The thymus gland has hormones associated with it. So thymosin is one, thymopoietin is another, and those are involved in processing the T cells into their separate categories. Again, this is really complex stuff. The T cells reproduce there, they go through several stages of development, and you come out with a whole bunch of subpopulations of naive T cells, if you will. It turns out that actually a lot of T cells are removed. And this is because some T cells, that are called cytotoxic T cells, have the ability to kill other cells. You do not want to have a system within your body that could find other cells within your body and destroy them. So remember when we were talking about bacteria having restriction enzymes that can chop up DNA, but they have to protect their own DNA so it doesn't get chopped up? We have a protective system like that. We have the ability to produce these T cells, subpopulations of T cells, some of which can destroy other cells. You don't want to keep the ones that will destroy your cells, so they get removed. And it's a fairly large percentage of them. About 98% of the T cells, the various T cells that are produced, are taken out, it's like right in the beginning, get rid of those guys, because they'll kill you. If you don't get rid of them, you end up with autoimmune disease, where they are actually causing damage to your own body. It's not okay. This is an interesting diagram because it shows a hematopoietic precursor. So this is a cell coming out of the red bone marrow, and it shows it going up in this long line of variation, and then it breaks into these two types of T cells. So you end up with T cells that have a marker on the surface that's called CD8, and over here you have another category that has a marker on the surface called CD4. The CD8 ones are the ones that destroy cells. The CD4s are the helpers. That's as far as I'm going tonight. Okay? We do have a quiz on Tuesday. Yep. Yep. So microbial control up to this point. It's a lot of material. And we'll take off from this point. I was trying to catch up a little bit in lecture. Caught up a little bit. Anyway, so, like I said, we're running that marathon. We're getting close to the finish line. We really have to put on that last spurt of energy. You guys also have a lab quiz this evening.
Is this our last one? Last lab quiz? Yep.
Oh my gosh.
It is.
Probably.
Yes.
Yeah.
Yeah, because then we have.
I mean, for short, one lab quiz because we only got up to 9.
That's a lot.
Right.
Yeah, Thanksgiving, planning.
Yep. Yep.
That means it's until he's our last song too.
Okay.
Right? No, we did a lab exam and then we had a lab. No, we did a lecture.